Good afternoon, everybody. My name is Nick Freemantle. I direct the Comprehensive Clinical Trials Unit at UCL. Today's speaker, Hakim, is our head of statistics there. Hakim joined us, I suppose, some months before lockdown with a particular interest in early phase studies, and suddenly has found himself with a rush of activity around COVID-19. So he's extremely well placed to deliver today's talk. So it's with great pleasure that I introduce my colleague, Dr. Dehbi, to talk for about 40 minutes, after which time we'll have an opportunity for some discussion, which of course you can add your questions on the Slido. Thanks very much. Hakim, over to you.
Thank you very much for this introduction. I'm going to start sharing my screen and start the presentation. So the title of this talk is The Story of Two COVID-19 Trials at the CCTU. CCTU stands for Comprehensive Clinical Trials Unit at UCL. It's really a pleasure for me to give this lunch hour lecture. I'm a big fan of the lunch hour lecture series. This is the very first one that I attended, and I've managed to trace it back via my calendar and the website of, or the webpage on YouTube of the lunch hour lecture series. So the title, and that was January 2012, was What Has Facebook Done to Us? And this topic is still as relevant as ever, given what has happened in the world and in the USA, for example, in recent weeks. I don't think that Zoom existed at that point in time, so the format was very different from the one of today. But I'm still very happy to give this talk today. So let's look at the outline of the talk. Firstly, just a few words on who we are as a trials unit at the level of UCL. Then I will talk in some details about Coronation, which is a prevention trial testing the MMR vaccine against COVID-19. Then FLAIR, which is our early treatment trial testing two antivirals in a factorial trial design. And I'm going to explain what factorial trial means. And then obviously at the end, a few comments, final comments and remarks. Okay, so the CCTU. We are part of the Faculty of Population Health Sciences, itself part of the School of Life and Medical Sciences within UCL. We're part of the Institute of Clinical Trials and Methodology. And in the real world, pre-pandemic, we used to live in this very fancy building on the second floor near Holborn in central London. Great office, great location, and I really miss it. And I do hope that we will be able to go back to the office in the near future. We're a small unit. It was created in 2011, so relatively young. We are 45 FTEs approximately, and we've got lots of different studies in various disease areas. We've got autoimmune disease trials, multiple sclerosis, obstetrics, cardiovascular disease, ophthalmology, and we cover all phases from dose finding to large confirmatory phase 3 or phase 4 trials. So that's it about us. This is who we are as a trials unit. Now, let's talk about Coronation. It stands for COVID-19 Research Outcomes Worldwide Network for Coronavirus Prevention. It's an incredible acronym, right? So let me tell you about the story of Coronation. So this takes us back to March 2020, 11th of March 2020, 4:35 p.m. GMT time. The WHO declares that the novel coronavirus outbreak is a pandemic. On the 17th of March, 6 days later, we have our first Zoom call with some colleagues of us at the Faculty of Medical Sciences at UCL, colleagues in the USA at Washington University in St. Louis, and in South Africa, University of the Witwatersrand. I hope I pronounced that correctly. What did we know then? There was this report published in February 2020 by the WHO China Joint Mission on COVID-19. And this figure 5 of this report, really caught our attention. It describes the trajectory of COVID-19 patients as a function of their health status, if you want, at onset. So as you can see, and we all got to know this over the last few months, the trajectory is quite binary. It's either recovery or death. And unfortunately, we've now discovered also that there is a category in between, which is called long COVID. I've read this report again, actually, while preparing this talk, and I think there are two key extracts that perhaps the world didn't take notice at the time. Firstly, it says that the COVID-19 virus could cause enormous health, economic, and societal impacts. The second main point is that much of the global community is or was not ready in mindset and or materially to act to contain the COVID-19 virus. So that's one thing we knew. What else did we know? That effectively, we had this report from China published in the New England Journal of Medicine in February with almost 1,100 participants. And the median age of these patients from various regions in China with COVID-19 was 47. So it's not as old as we tend to think, 40% female, and 6% of these patients either ended up in ICU, reached mechanical ventilation stage, or died. This was the state of the knowledge back in March 2020 when we jumped on our first Zoom call with our colleagues around the world. What did we decide? Obviously, we said, we are a trials unit, and we said, let's do a trials, right? Let's do a trial. What are the three main questions to answer if we wanted to do a trial? The what, the who, and the how. So the what kind of trial? Should it be a prevention trial, early treatment, or late treatment? What treatment, what intervention should we choose? When it comes to the who, should it be in the general population or specific subgroups of interest? The how, should it be placebo-controlled? What study design can we choose? Where are we going to do this trial? How can we collect the data, especially given the current circumstances? How to administer the treatment? So these were all the questions that we had to answer. And I'm now going to take you through, how we try to answer all of these questions for Crown. So let's start with the what. What phase, what treatment or intervention? And this is now the story between March and June, July of last year. What we had in mind is the importance of prevention. Benjamin Franklin is known to have said, an ounce of prevention is worth a pound of cure. If you think about the process of COVID-19 and the amounts involved, this is obviously extremely accurate. Pre-exposure is much cheaper than treating patients. And treating patients who have reached hospital stage or ICU is even more expensive. So we had this mindset in mind. At that point in time, drug repurposing was the main approach. There was this very helpful article in Nature Biotechnology from February 2020 with the title, Coronavirus Puts Drug Repurposing on Fast Track. And they had this table, super helpful for us, of various drugs that had been tried already in China in various clinical trials. This table, interestingly, includes remdesivir, which turned out to be the first drug approved by the FDA in the US for treatment. And the last row of the table, chloroquine or hydroxychloroquine. And chloroquine was our first choice for the study, for Crown. Now, obviously, you want to know why chloroquine? What early evidence did we have? Well, it was known to decrease viral replication. It was known to treat pulmonary diseases. And at that point in time, coronavirus was known as a pulmonary disease in the first place. It treats pulmonary hypertension, pneumonia. It prevents cytokine and chemokine storm. There were some evidence of this. And there were evidence also that it may prevent viral entry into cells. It's a very successful drug. I mean, chloroquine has an extensive track record against malaria, and it's available easily and worldwide, and it's not expensive. So as a prophylaxis treatment, prophylaxis means prevention, as a prophylaxis treatment, it did make sense. So what were the objectives of Coronation version 1 at that stage? What did we have in mind? We wanted to demonstrate, firstly, that chloroquine is superior to placebo. We wanted to observe a reduction in the proportion of participants in the trial becoming symptomatic with COVID-19 if they were in the chloroquine arm compared to the placebo arm. There was another question of interest, whether a medium or a low dose of chloroquine could be non-inferior to the high dose. From a more operational perspective, we wanted to progressively allocate more and more participants to an active arm. Now, in terms of design, we were going to do a large international placebo-controlled trial with up to 55,000 participants. We had 4 treatment arms in mind: a placebo arm, a chloroquine low dose, weekly tablets, medium dose twice weekly, and a high dose arm with daily tablets of chloroquine. The primary endpoint was going to be incidence of symptomatic COVID-19, and we were interested in a Bayesian design with frequent interim analysis for efficacy, futility, or harm. We had obtained funding from the COVID-19 Therapeutics Accelerator. We had obtained 9 million for Coronation. We were very happy. I remember the day we heard the news. It was a feeling of joy, really, that we were going to do something about the pandemic. For those of you who don't know the COVID-19 Therapeutics Accelerator, it's a group of funders. It includes the Wellcome Trust. It includes the Gates Foundation, Bill and Melinda Gates Foundation. It includes the Dell Foundation, the Avast antivirus foundation as well. I don't know if they have a foundation, but I've remembered that Avast is part of this because Avast is an antivirus company for computers and laptops, etc. And it also includes famous donors, including Madonna. We even tweeted Madonna at the time. We tweeted Madonna to thank her or her organization, but they never replied, which is rude. But anyway, so we had 9 million to run Crown, and it never happened. Crown never happened for 3 main reasons: because of twice impeached Donald Trump, because of the Lancet and the NEJM fiasco, and because of evidence from post-exposure trials that started to emerge that show that chloroquine may have low efficacy to avoid symptomatic COVID-19. So I'm going to talk about these 3 reasons now. Firstly, twice impeached Donald Trump takes hydroxychloroquine out of the blue, and that was April, if I remember well, or May of 2020. At that point, science had been replaced. Hydroxychloroquine was unproven to have any benefits for patients. There were obviously in vitro evidence that it was able to inhibit replication of the virus, hydroxychloroquine, but there was no clinical evidence at that stage. Hydroxychloroquine and chloroquine are associated with rare cardiac side effects, but Donald Trump decided to take this drug as prophylaxis. We knew that this would add additional challenges for us in terms of recruitment because of all of the media attention for recruitment for our future trial that was about to start. People would think, if Trump takes it, then most probably I should not take it. Or if Trump takes it already, then why do we need a trial? That was the first main reason. Then there was the Lancet and the NEJM fiasco. They published studies based on a data set that was produced by Surgisphere, which is a company that includes a sci-fi writer, among others. And these papers showed, based on fabricated data, that hydroxychloroquine or chloroquine were not efficacious prophylaxis or treatment interventions for COVID-19. At that point in time, governments and asked for some COVID trials to be temporarily paused. Now, we knew at that point in time that this would be very difficult for us to recruit. The drug, effectively, chloroquine, hydroxychloroquine, had lost a lot of credibility in this big fiasco. And I'm using the word fiasco because in the end, what happened is that these two papers were retracted. The authors have recognized that they never had access to the raw data. And turns out the raw data had been fabricated. And now the third reason why it didn't happen, which is probably a bit more scientific, is that some evidence started to emerge from post-exposure prophylaxis trials that there was no difference between chloroquine and placebo for prevention of COVID-19, the development of the disease, for people who had been exposed to the virus via their contacts. And that's why we call these studies post-exposure prophylaxis. They were contacts of what we call index cases, so people who have tested positive. And so these people we knew or were known to have been in touch with positive cases. And they were given chloroquine or hydroxychloroquine as potential prophylaxis, but it turns out that after a certain period of follow-up, there was no difference between the groups, chloroquine versus placebo. So effectively, we were back to square one. So now let's go back to today. We still had prevention in mind, right? I mean, 1 ounce of prevention is worth a pound of cure. However, the thinking had evolved and we were considering vaccination. Why is that? Simply because it's much more practical as a prophylaxis treatment. You just need to take 1 injection or 2 injections, as we know, depending on the vaccine maker, and then you can continue your daily life. If it was tablets like chloroquine or hydroxychloroquine, then people would have had to take these tablets every day or twice a week, etc., which is much more cumbersome. So our thinking had shifted towards vaccination. We chose the MMR vaccine. Why is that? Because obviously, firstly, we are not a big pharma company, so we are not in the business of developing a specific vaccine. But there were, interestingly, immunological as well as epidemiological evidence that the MMR could have a role to play in the pandemic. So firstly, immunology. We know that live attenuated vaccines, like the MMR, may train our innate immunity, our ability effectively to fight infections in general in a non-specific way. We also know that the specific antibodies that are produced by the body when it is faced with the live attenuated vaccine, the live attenuated viruses effectively in the MMR vaccine, these specific antibodies may have cross-reactivity to SARS-CoV-2. Cross-reactivity means that they could also bind to SARS-CoV-2 because the spike proteins of these viruses are not too different. From an epidemiological perspective, we were then in July of 2020, and there was some evidence already that there were less cases of COVID-19 amongst people who had recently received an MMR vaccine or a BCG vaccine. And in the past, there have been evidence that the rate of various infections and diseases had gone down after the MMR was introduced in the country. Now, let's look at the immune system. it effectively, I am simplifying everything here, but it has two main pillars. The innate immunity on the one hand and adaptive immunity on the other hand. So innate immunity, what is it? It works with recognition of pathogens via pattern recognition receptors. So it's non-specific and it involves reactions such as phagocytis, cell locomotion, or cytokine production. Adaptive immunity is completely different, and it involves specialized T and B cells. It's a slow process. It involves our lymphocytes, so white blood cells, and it is antigen specific. Effectively, we're talking about our long-term immunological memory. It's interesting to note that our innate immune cells can show also adaptive characteristics. So the specific vaccines that have been developed in recent months, including the Pfizer vaccine, the Moderna vaccine, the Novavax vaccines, and all of the other vaccines, they are effectively exploiting the adaptive immunity side of our immune system. And we then thought that we could actually focus on the other side, on innate immunity. So let's talk a bit more about immunity, trained immunity in particular. Here, firstly, this is how it works with a traditional vaccine with our immune memory. So this is the trained, the adaptive immunity. So we give the traditional vaccine, it activates our adaptive cells, for example, the Pfizer vaccine, and the antibodies from the B cells and the T cells either block the infection or kill the infected cells. However, if we were to give the BCG or the MMR vaccine, what would happen is that our innate cells, the other side of the immune system, the innate cells, would be pre-activated, would be warmed up, if you like. And what would happen would be that monocyte cells would actually be able, hopefully, to engulf and kill the virus. When it comes to the coronavirus infections, how would it work, in practice, in time? So we would give the MMR vaccine, and this would create an innate immune response. It would, activate our immune system, the innate side of it. We would then, or the participants would be infected with the coronavirus, potentially. And what would happen then is that we would see an increased response. It would be boosted thanks to the fact that the immune system would have received a prior vaccination prior to the infection with COVID-19, with the SARS-CoV-2 virus. So that was the trained immunity side of things. Now, innate immunity side of things. Now look at this slide, and this is now with respect to adaptive cross-reactivity. We've got the spike proteins of the measles virus that are not too different in shape. There are some similarities with the spike proteins of the SARS-CoV-2 virus. So the antibodies that are generated by the measles vaccine may also bind to the SARS-CoV-2 virus. So we had these various pieces of evidence that the MMR may play a role in the pandemic. And that was the what. This was our treatment. Let's now look at the how. What are we going to do? We want to test, as I have said, whether the MMR can prevent COVID-19 or decrease its severity. So the trial is very traditional and very simple. It's a 2-arm parallel trial where participants are registered and screened for inclusion-exclusion criteria and then randomized 1-to-1, which means 50-50, between the MMR and the placebo groups. We would follow them up for 60 days, and our primary endpoint is PCR confirmed COVID-19 with symptoms. In terms of sample size calculation, how many participants do we need to demonstrate that the MMR has an effect, if we assume that it has an effect? we are going to recruit 5,000 participants in total, 2,500 per arm. And on this plot, you've got the sample size calculation that we have done via simulations. And I'm going to explain how we did it. Firstly, what is plotted? It's the probability to declare efficacy as a function of the sample size per arm. We've got three curves that correspond to three different event rates in the placebo arm of the trial. And by event rates, I mean the proportion of participants in the placebo group who would develop COVID-19 during the course of follow-up. And it's important to remember that these participants, they are COVID-free at the start, but some of them would actually be infected and develop the disease during the course of follow-up just because of their daily activities. The probability to declare efficacy, it's defined in a Bayesian way. We want to demonstrate that the probability that the odds ratio, and I'll explain what the odds ratio is, we want to know that the probability that the odds ratio is less than 1 is greater than 95%. So as I said, there are multiple inputs into this calculation, but we calculated that we needed 2,500 participants in total. And this is based on an expectation that the odds ratio would be at least 0.7. So let me now talk about the odds ratio. Firstly, it's important to note that many clinical trials have a similar design to what I just showed, right? We've got two treatment arms and a binary endpoint, yes, no. In our case, yes, development of COVID-19 or no. So effectively, we can summarize the results in a 2x2 table. So here we've got our 5,000 participants. These are hypothetical numbers. Obviously, we haven't finished the study, but these are our 5,000 participants broken down 50-50 into MMR and placebo. And we would have here, hypothetically, a 10% event rate. 500 out of 5,000 people would have developed COVID-19. The breakdown of these 500 is 150 in MMR, 350 in placebo. If there was no effect of the treatment, you would not expect to see an imbalance. You would expect to see 250 participants in both study groups. It's not the case in this hypothetical example, which would imply here that perhaps the MMR is indeed effective at reducing COVID-19 as a prevention trial, as a prevention treatment. So now, what is the odds? The odds is the ratio of people who do and who do not have the event in the periods of interest or 60 days after receiving the MMR. In the example that I had in the table, we had 150 participants out of 250 who developed the disease. So the odds are 150 divided by 2,500 minus 150, which is 0.06. And these are the odds of developing the disease in one treatment arm. Now the odds ratio is just the odds in the treatment arm divided by the odds in the placebo arm. In this case, it was 0.39, this odds ratio. We do not expect to see something as dramatic as this. We just hope that the MMR is going to provide an odds ratio less than 0.7. And if it is the case, if it is true that the MMR has an effect on the coronavirus, then 2,500 participants in both study arms should be enough, should be sufficient for us to see this in the data. As I said, it's a Bayesian trial. So in Bayesian statistics, what we do is that we put distributions on the quantities that we try to estimate. And in this case, it's the odds ratio that we try to estimate. So on the left-hand side here, what you have on the slide is the distribution that we have applied on the odds ratio prior to the start of the study, with 50% of the mass less than 1 and 50% of the mass greater than 1, which effectively means that, you know, we don't know whether it is going to have an effect. And then in the Bayesian approach of, or in the Bayesian mechanics, if you want, you combine your data with your prior on the odds ratio to derive a posterior distribution for your odds ratio. And as I said before, what we would like to have is a shift to the left of this distribution where 95% of the mass would then be below 1, which would mean that we have 95% certainty that the odds ratio of the MMR compared to placebo is less than 1. Let's now look at the key operational feature of the trial. It's still the how, right? I've just described the study design and the sample size calculation. It's part of the how. We also need to consider how effectively we're going to collect the information that's going to come into the calculation. This trial is a virtual contactless trial, obviously, given the situation. So it would involve self-reporting of the eligibility by the participants as well as their adverse events and their outcomes. People in remote areas, it's an international trial, not only in urban centers, would then be contacted by SMS every week or so in order to collect information on their health, whether or not they are healthy or if they have been diagnosed with the disease. And these SMSs would then feed into the main trial data sets. Achieving this sort of virtual contactless trial from an operational perspective and database perspective is a gigantic challenge for us. And it has been a success, and we've collaborated with Telerevit and other SMS companies that have helped us. Some of these companies have helped us for free, given the situation, and we are very grateful to them. And lots of work from all of our operational colleagues has gone into the contactless nature of the trial. Let's now move on to the who. Who are we considering for the study? Initially, we had in mind healthcare workers. It was known that there was a high incidence and some severe case of COVID-19 amongst healthcare workers, because obviously they are exposed to sick patients. However, via a protocol amendment, we have progressively expanded the eligibility to key workers as well as other adults at risk of exposure to SARS-CoV-2 due to their work. So now we are effectively recruiting in South Africa, among others, any adults at risk of exposure to the virus. It's not only healthcare workers anymore, but obviously healthcare workers are still part of the potential pool of participants. The study is ongoing. The study is live. Here is our website. This is where everything starts from the participant perspective. They click on check your eligibility, and then they go through the motions. It's enrolling. We have enrolled participants in the US, in the United Kingdom, and now we are focusing on the other countries, which include Zambia and South Africa currently, and we hope very soon to start recruiting in Zimbabwe and in Ghana. So that was the story of Crown so far. We are recruiting. We hope to be recruiting rapidly over the next few weeks and months, and we hope to have results by mid-2021. Let's now talk about FLAIR, which is the second trial of COVID-19 in our department. So what does FLAIR stand for? Favipiravir plus minus lopinavir, an RCT. RCT means randomized control trial of early antivirals. So I'm taking you back now to March and April 2020. What did we know at that point about viral infections? As I said before, drug repurposing is the first option when faced with a new virus. If someone is infected in general, not only for the coronavirus, too, early treatment gives best results. Combination treatment, think of HIV, dual or triple therapy, tend to work and tend to work well. And dose optimization is an important topic. It might be important that to give higher doses for COVID-19 than with other diseases. In terms of drug development, we in general separate the preclinical stage to the clinical stage. And in both stages, we go from learning to confirming. In the preclinical stage, from in vitro to in vivo studies, and then in the clinical stage, from phase 1 to phase 3 trials. FLAIR is a phase 2 trial, so it's somewhere in the middle between learning and confirming, but more leaning towards learning here. And learning often is embedded in PK/PD models, pharmacokinetics, pharmacodynamics, which means what does the drug do to the body and what does the body do to the drug. So what did we know at that point in time, March, April, from PK modeling in vitro? It was known that obviously antivirals needed to be started early, within 5 or 7 days of infection. Hydroxychloroquine, as well as lopinavir, ritonavir, a HIV therapy, and interferons as monotherapy would have perhaps 30 to 60% inhibition of replication. Now, remdesivir was predicted already from in vitro models to achieve 87% inhibition. Interestingly, there is a drug called Favipiravir that has the same mechanism of action, but is available as an oral formulation instead of intravenous. So this is the focus of FLAIR. We are focusing on lopinavir, ritonavir, and Favipiravir. These are the two drugs that we are testing in the study. What clinical evidence did we have at that stage? For Favi, firstly, Favipiravir, it's a drug that is active against a broad range of viruses, including yellow fever, Ebola, and influenza. It's mostly an influenza drug, though. As I said before, similar mechanism of action as remdesivir, but oral formulation. So much more practical again, especially for early treatment when people are not in hospital. Shown in vitro to be active in terms of inhibition of replication. And there was very little data available, but there were claims from Chinese authorities that of moderate effectiveness for patients treated with Favipiravir. There were a few news reports or articles published already in March 2020 about Favipiravir as a potential treatment for COVID-19. What evidence did we have for lopinavir, ritonavir, which is this HIV therapy? From an in vitro perspective, lopinavir is known to have some action against coronavirus. And then there was this study by Cao or Cao, I don't know how to pronounce, et al. In the NEJM with 199 patients in early 2020 in China. And they were randomized between control or lopinavir, ritonavir. And there was no difference, as you can see on the plot, in terms of time to improvement. However, in a post-hoc subgroup analysis, there was a small difference in mortality for the participants who were treated within 12 days after the onset of symptoms. So effectively, this was the clinical evidence that we had for both treatments. So now let's move on to the how. Study design-wise, we thought we would do a 2x2 factorial trial. What does it mean? So there are effectively two randomizations because there are two treatments. The first randomization for the first treatment, say lopinavir, is between placebo or lopinavir, right? And then let's look at the participants on the right-hand side who would have been allocated the placebo treatment of lopinavir. They would then be randomized to either Favipiravir, the other drug, or the placebo of Favipiravir. And it's the same reasoning on the left-hand side for the participants randomized to lopinavir. They would be either randomized to the placebo of Favi, or they would also receive Favipiravir. effectively, we've got a quarter of the participants that receive both drugs. It's the left-hand side box. A quarter of the participants receiving 2 placebos. And then we've got participants who receive just one of the 2 treatment drugs. Now, there is another way of presenting this data. It's in the format of a 2x2 table again, where you've got the control participants, who are the participants who don't receive any of the 2 treatments. And then we've got the participants who receive A plus B, and some participants who receive just A, some participants who receive just B. Now, what are the questions, the research questions of interest for FLAIR? Firstly, does lopinavir reduce viral load in the upper respiratory tract compared to placebo after 5 days of treatment? Same question, but for Favi. So we are effectively interested in viral load and how the viral load evolves with the drug after 5 days of treatment. And then an important question, a key question, is, do both treatment interact synergistically? So do they complement each other, if you like? Like, do you get an additional boost on top of the effect of Favi and on top of the effect of lopinavir by giving both treatments together? So this is what we called synergy. So the primary endpoint, as I said, is the viral load in the upper respiratory tract. This graph actually shows the sort of data that we will have. Effectively, we'll have the viral load at day 1 for patient 1 in blue, and their viral load at day 5. Same thing for patient 2. We've got their viral load at day 1 and their viral load at day 5. And as you can see here, it is expected that with time, the viral load will decrease. Right? One could say here that obviously the viral load has decreased more markedly for patient 1 than patient 2, but it has decreased for both, right? And this is at the patient level. Now, if we look at the data or at the trial itself at the group level, right, what do we expect to see at baseline? Via randomization, as I said, this is a randomized trial, the groups are expected to be similar at baseline in terms of viral loads. We don't expect to see massive differences between the group that has received 2 placebos and the groups that receives the 2 drugs at baseline, right? And what we hope to see is that thanks to the treatment, the viral load will decrease more markedly in the Favipiravir group. The straight lines that we have here represent the 95% confidence intervals around the mean values calculated for the groups, right? And what matters is to see a difference at the end. We have powered the study in order to identify a 0.9 log10 difference on the viral loads. And to give you an order of magnitude, a 1 log10 corresponds to a 10 times increase in copies of the virus per ml. Sample size-wise, we are going to recruit 240 participants, 60 participants per group, and this gives us 90% power to detect this difference that I mentioned, 0.9 log10, for the main effects. What do we mean by main effects? The effect of one drug compared to placebo, and in addition, we've got 80% power for the interaction to detect a difference of 1 log10 difference by giving both drugs together on top of the main effects. Let's now move on to the who. Who are we recruiting? Well, effectively, anybody, adults, obviously, in their first week of illness. The trial is currently recruiting. It's recruiting in and around London. So if anybody in the public is in this unfortunate situation, do get in touch with the Royal Free Hospital, Dr. David Lowe. They will come to your place. They will give you the drugs that you have been randomized to. The study is effectively currently recruiting, and it also applies if you know anybody who is in this situation in or around London. Final comments. the last part of my presentation. I want to talk about the importance of these two trials. In my opinion, obviously I'm biased, but they are very important. Innate immunity, it's the second pillar of the immune system, right? Obviously, the specific vaccines have shown remarkable efficacy, but they've exploited the adaptive immunity part of our immune system. The other pillar is available. We need to make the most of it. It might even be that innate immunity trained by the MMR or the BCG vaccine may boost the efficacy of specific vaccines. This would be the topic of another trial. We would need to raise funds if there is, if there are funders in the audience or rich philanthropists. Do get in touch with us. We are very interested in this question. And otherwise, the MMR and the BCG are very well-established vaccines, right, with very well-known safety profiles. So for future pandemics, they could be reused as first line of defense while we wait for the development of other specific vaccines. But thank God, the MMR, the mRNA technique has shown that vaccines can now be developed very fast. Now, for flare, why is it important? Firstly, vaccines do not have 100% efficacy. So there will be people who will get sick. Some variants, we may have some surprises with the variants. Some variants may escape the specific vaccines and our innate immune responses. And it's better to have early treatment than to find, I mean, it's super important to find treatments that work in late stage disease, but it's even more important public health wise to find treatments that work at the early stage of the infection. Second comment that I want to make, these studies, they are gigantic collaborative efforts. Dozens of people are involved, including some of the names that I have mentioned on these slides. And there are many, many more people who I have had the chance to work with. It has been a pleasure over the last 11 months now, I think, almost, or 10 months. Thanks to the funders, thanks to the COVID-19 Therapeutics Accelerator, who have now allocated, I mean, up to October 2020, $98 million in grants. Thanks to LifeArc, a UK charity that has funded FLAIR. And this is the end of my presentation. I'm not wearing a tie today. I'm not a black cat. But if there are any questions, I would be very happy to answer. And thank you very much for the attention.
Thank you very much for the presentation, Hakim. That's you've done a lovely job of covering a lot of ground. We do already have a couple of questions in the chat. A very relevant one to start off with: information and misinformation. Jill's asking the question: to counter pernicious misinformation, could all COVID-19 trials and all COVID-19 mass vaccination programs publish yellow card data on adverse side effects? Did you get that?
Yes, I heard the question.
So I guess the question is really about transparency. So, and transparency and trust in clinical trials is such an important thing. So do you have any thoughts? Have you any learnings on transparency from the COVID-19 era?
Yes, absolutely. So I think it's a great point. It's very relevant, especially in this era where we've got vaccine hesitancy or vaccine skepticism. It's a great thought that we should, as much as possible, publish our side effects, as well as the adverse events that we see in any of the clinical trials. And from my perspective, it might also be an idea to publish data on side effects from the vaccine rollout programs that are currently taking place around the world. Lots of people have been vaccinated already in the UK as well as in Israel. So it would be good to know and to have details around these side effects. Obviously, the main difficulty here is data protection. right? Data protection and security or safety of or possibility of re-identification of the participants. No one wants to know that. No one wants to be in a position where they can be re-identified that they are the participant who experienced that very bad side effect in, say, the Pfizer trial. It is known that the Pfizer trial was stopped at some point for adverse events, but there are very few adverse events in those trials. So it might well be that by making the data public, there will be a chance of re-identification. So we need to balance these various aspects, but overall, we have to do all we can to counter vaccine skepticism and hesitancy.
Yeah, and I think if there's a vacuum, it's clear that that's very quickly filled with other issues. So, you know, when the Pfizer BioNTech vaccine was first being implemented in the UK, first being used in the UK, I had colleagues in the NHS describe, very high rates of fever and things, 30% of people experiencing fever after the vaccination, which just wasn't seen in the large exposure in the randomized trials. And actually, in truth, wasn't going on in the NHS either. But, you know, people's anxieties and expectation led these sort of these stories to go around, and you know, we're surrounded by information and misinformation. So I think this is a really important agenda. Another question from Anonymous this time. For Crown, are new arms planned to be added? Some trials labelled as adaptive platforms have just been a series of 2-arm trials. Can I ask your thoughts?
That is a possibility for Crown. Crown is a platform trial. We are starting with the MMR with 5,000 participants. And depending on how things go and where the pandemic goes in particular, we might add additional treatments afterwards. It might be that the MMR will have been shown to be effective, and if that is the case, the MMR would become the backbone treatment, and we would add additional treatments on the platform, perhaps even a combination of a specific vaccine versus and or with or without the MMR, for example. That would not be the case if the MMR becomes the backbone treatment. Another option, for example, would be a molecular mask in terms of prevention. I know that this is currently developed in the School of Pharmacy at UCL. So we could combine those treatments either as parallel arms or factorial trial events in the same way that we've done for FLAIR. It could be that the trial will stop after the first intervention, and it will depend on the finances. It will depend on the state of the pandemic and whether it is still relevant at that point.
Yeah, I think the point about combination treatments, I mean, when we look back at the success of therapies for HIV and AIDS, it has been combination therapy which has really led the way there. So, I think the idea of examining that flexibly, and that's personally why I'm so enthused by the FLAIR trial, because it's beginning to look at using those kind of learnings and taking them into this area. Jill's got another question for us. Many older people are minimizing outings and external contacts to minimize infection possibilities. How do trials adjust to prove accurate effectiveness? And of course, the attack rate in the Pfizer studies, the Pfizer study, and actually in the AstraZeneca Oxford study, was really very low, with a very low number of events. So what can we do? And AstraZeneca particularly only included younger people. So what can we do about that, Hakim?
Excellent question. Excellent question. I am not sure that I have got. I've got obviously the answer for this. I can think of various ways to approach this. Firstly, large phase 3 trials, it's only part of the big picture, right? There are phase 1 or if not phase 2 trials, and there are also in vitro and in vivo studies that are done prior to the large trials. So they are just one piece of the puzzle. If all the people are not part of the large trials, at least in the first instance, it doesn't necessarily mean that the vaccine is not going to work on those populations. It is just the case that initially they were not included in, say, the AstraZeneca trial, but they had preliminary evidence from PK/PD data as well as phase 2 trials that the vaccine was effective on older participants. So that's one thing to note. With respect to the fact that the attack rate is very low and that older citizens tend to minimize outings, it's a fact. It's a fact, and I don't think that we can think about it. Go and take the tube just to see if they catch the virus. So it's a very complicated. It's an excellent question.
I think I would add just
really know at this stage what we can do.
I would add just one extra point. We're in the situation, you mentioned phase 4, we're in the situation for some phenomenal natural experiments. So the state of Israel, for example, has achieved a very high initial rate of vaccination. And many of us are watching the progress of the virus there with great interest now, because that shows where we can be a larger country with greater logistical challenges to achieving high vaccination rates in a few months' time. So I think we can. The trials, as you say, are part of the story, but we move on to to seeing what actually happens in the real world to hopefully confirm there as well.
Very much.
Overall,
absolutely. I was just going to add actually
the question.
Oh, sorry.
Go on.
No, carry on. No, sorry.
I was just going to ask a general question.
So
We've got a few minutes left. The general question comes to my mind. I think that we've learned a fantastic amount in the last months in the context of trials, in the context of this pandemic. What do you think those key learnings have been?
It's a very good question, I must say. It's a very broad question also. Key learnings. Firstly, I think it's really important to do the homework before starting a trial in terms of, in vitro evidence, in vivo evidence potentially, and whether small trials have shown any signals, right? In the end, we didn't do the chloroquine version of Coronation, and had we rushed into it, we would probably have wasted our time and the time of the participants. It's probably better to do more in terms of preclinical data as well as small phase 2 trials prior to jumping to large phase 3 trials. We have also seen that in the late stage treatment trials with the recovery trial, for example, where quite a few treatments have actually not shown any benefits, right? Including chloroquine, if I remember correctly, in late stage COVID-19. So that would be learning number one, that we definitely have to do our homework before launching into large phase 3 trials. Second learning, the influence of external factors on these trials, right? What has happened with the Surgifer fiasco, NEJM and the Lancet, these sort of things are extremely counterproductive and should never have happened, but hopefully these sort of mistakes won't happen again in the future. And I do hope that these journals that are supposed to be top journals in the world have adjusted and obviously have learned from this mistake. I'm not saying that mistakes do not happen, but hopefully they have learned from it. Another learning is that it's really challenging, logistically speaking, to develop a new trial or a new platform in the space of a couple of months, especially for trials you need that are for which this is not a specialism, right? We are not Pfizer, effectively.
Excellent points. Now, I notice that it's got to 2:00. We're all slaves to the clock. It just remains to me to thank you, Hakim, for an absolutely excellent and stimulating talk. Thank everybody for joining us today. Please look out for the results of these studies. I'm pretty confident that they're going to get exposure in the national press, and I'm sure they'll be distributed amongst through more conventional UCL lines. Best of luck with the rest of your day, everybody. Keep safe, and thanks very much for joining us today. Thanks again, Hakim.
Thank you, everybody, for listening. Have a good day.